All right, thank you, Arjun, for the introduction and the invitation. And of course, it's great to follow up on what Aaron talked about in terms of looking at nuclear structures. And so, in textbooks, we get these pictures of chromosomes that are organized in different orders of magnitude. So they're, you have the double helix, and then you sort of wrap around histones, organized into this fiber, and so on. And then, for us microscopists, when we look at the cells in the microscope, and these are mouse embryonic stem cells, you get an image that looks like this. You have some DAPI bright spots, so you have a bunch of empty regions and, the rest of the nucleus. So how does the chromosome structure, right? How do you reconcile these two pictures? How do you see them both in the same cells? And so there are currently two approaches to looking at chromosome structures in cells. Aaron touched on some of this already. So one of those approaches is using these chromosome capture type of experiments, whether with Hi-C or Sprite or GAM, to measure pairwise contacts or multi-order contacts between different DNA loci, to generate these contact maps and then reconstruct the structure of the chromosomes using computational analysis. And then, of course, the other approach traditionally has been using microscopy to image directly the chromosomes, and this is done with either DNA fish or chromosome paint, where you actually see the chromosome territories, individual cells, and more recently, done at higher multiplex by these chromosome paint, oligo paint methods developed by Ting Wu's lab, and most recently with in situ sequencing. But in addition to the DNA, we also want to look at the nuclear structures in cells in the nuclei. So there are many different types of nuclear bodies. So you have the nucleolus, where ribosomes are synthesized. You also have these cytochromatins, nuclear speckles, and all kinds of nuclear bodies. So how do the DNA and these nuclear bodies, and lanolin, of course, that Aaron just talked about, how do all these things come together, and how do we get this integrated picture of what the chromosome looks like? So I'm going to tell you about this experiment that we did recently, where we looked at both the RNAs as well as thousands of DNA loci, as well as a number of antibodies, all in the same single cell, and put all of this data together. And I should say, imaging-based methods is a very nice platform to integrate all this information rather than droplet-based methods, right? Because first of all, for methods like CySeq, it's actually hard to get the antibodies into the cells when they're still intact. So you can do this on surface proteins, but for imaging-based methods, it's very easy to get intracellular proteins, especially nuclear proteins, to be targeted by antibodies. Okay, so all of this method is based on single molecule FISH, which of course was pioneered by Arjun and Rob Singer. And this is a method that allows you to detect individual transcripts or DNA locus, with a set of probes, and they're labeled, and you can hybridize them in the cells, which then when you image on the microscope will give you these punctas. And so a couple of years ago, we developed this method called sequential FISH or seqFISH, where we use multiple rounds of hybridizations to encode different molecular species. So for example, RNA1 was labeled green the first time and labeled blue the second time. And so this color sequence gives you the identification for each of the transcripts. And you would design probes against all the transcripts you're targeting on, and then design the barcode associated with that. So there are other methods using the same kind of ideas, using sequential hybridizations, but with slightly different coding schemes in the literature. And we showed that this method can really scale up to the genomic level, not just level of tens or hundreds of proteins, mRNA molecules, but levels of tens of thousands. And the first demonstration was done actually looking at introns, and this is actually also following up Arjun's ICE FISH demonstration to target intronic regions, the nascent transcription active sites. So we're looking at a single cell here, where each one of these dots is a nascent site of synthesis of RNA hanging off the chromosome. And so that also gives us a sense of what the active chromosomes look like in individual nuclei. And, I should say that the blues are the DAPI-rich heterochromatic regions, and then the white are the nucleolus. And then this is where we were iterating through all the different chromosomes, so we ended up on the X chromosome here. And then a couple of years ago, Linus, who was an amazing graduate student in my lab, demonstrated that you can do RNA-seq plus to target 10,000 different mRNAs in single cells. And this is done not only in culture cells, but also in tissues. This is a brain slice. You can zoom in successively into this region of the cortex, and then zoom into the cell here, and zoom in again to see all the different transcripts that we can decode from this particular image here. And the plus here refers not to a lot of genes. It's not that seqfish plus can do more genes than seqfish. You can already scale to 10,000 genes with seqfish. The plus refers to this deterministic super resolution methods that allow us to resolve very crowded samples and see a lot of transcripts, because on average you have something like hundreds of thousands of transcripts in a cell, and they don't all pack into the cell by, if you just did conventional microscopy. Okay, so we apply the seqfish plus method to look at DNA now, so we can resolve the chromosome structures on the level of roughly about a megabase or so across the entire genome, and then observe that with super resolution, about 100 nanometer resolution in each of the experiments. And so you can see here, we have a bunch of rounds of hybridizations that gives us what we call a pseudocolor, and then the pseudocolor changes over multiple rounds now, and that gives us the identity of exactly which chromosomes we're looking at. So for example, let me back up. We can see some of the chromosomes, some of the loci correspond to one of the chromosomes, and then correspond to another one. And then that was done in one of the fluorescent channels. So we have two of those fluorescent channels that gives us a megabase coverage. But then we have a third channel that images just about 1.5 megabases, but at 25 kilobase resolution. So what we do there is we hybridize, these 25 kilobases resolution walk along the chromosomes. We do that in parallel for all 20 chromosomes at a time. And then, after that's done, you can then paint the parts of the chromosomes that you targeted. So you know that these dots, as you're walking along from region 1 to 60, belongs to chromosome 9, and these guys belong to chromosome 15. So from that, you can decode another 1,200 loci, and then know exactly, at super high resolution for these, for these short regions in each of the chromosome. And then on top of that, right, we can do the multiplex immunofluorescence. This is using an oligo-labeled antibody, so that you can hybridize the antibodies in a pool and then fix them in, and then read them out using exactly the same method that we read out the fish probes. So it's all integrated in a single experiment here. And you can see, going through the different types of antibodies. There's about 17 antibodies in this particular experiment. You can see the lamins show up in lamin regions. The heterochromatic markers labeled the heterochromatins, and nucleolar markers marks the nucleolus, and so on. And it's actually really essential for the protocol to preserve the structure of the nuclei very carefully. And we took a lot of care to show that even after heating, which is required for DNA fish, that the immunofluorescence basically overlaps almost perfectly. And this requires a number, a double fixation protocol. So you basically fix twice and then unfix one time, and then the structure is still preserved. So that's actually really essential for what I'm about to show you the rest of the talk. So here's a reconstruction of all the about 3,000 chromosome loci that we image in this particular cell here. And we're iterating through the different chromosomes. You can see many times there are two separate alleles, but then sometimes they're fused together into a single region in the nucleus. And then after all the chromosome imaging is done, then we can look at the antibodies. So they form these discrete globules. Many of them form discrete globules inside the cells. So those are the inactive marks, and these are the active ones, RNA polymerase, acylation mark speckles, lamin. And then now we're looking at the repeat regions in the genomes. So we can do all of that in the same nuclei. And this is work done by this fantastic graduate student at UDI. And I would say, I was really UDI's grad student for the last year during the pandemic, because UDI generated data, and we were looking at the data together, analyzing it, and it was just really fun to do that when we were all stuck at home. So going back to the title slide, right? We have this image of the DAPI. Now we can see exactly what the chromosome looks like inside each. I mean, this is a one-to-one correspondence of the DAPI and the chromosome images. And, we see what the chromosomes look like. And in fact, we know that the results are very good because by comparing to the seqFISH Plus data with the Hi-C data, shown on the upper right versus lower left, they match very well at different time length scales. At either the entire chromosome scale, you see the 20 different chromosomes in blocks, or at the megabase for 25 kilobase resolution. And so if you zoom into that one cell there, you can see the different chromosomes, the chromosome 5, the two alleles, and you can draw a line through all the loci to see what it looks like. Then you can zoom in further into that 25 kb resolution and then see that high resolution. And you can easily spend half a year just staring at these structures. They're really beautiful, but it's actually very hard to say anything about them because they look very random in individual cells. They're highly, highly variable. So how do we get this sort of dimension reduced picture of chromosome organization? And here's just where the antibodies, immunofluorescence becomes important. So, in the same cells as you've seen, you can look at the antibody stains, and you can here see the heterochromatic marks, the H3K9 trimethylation, the DAPI, and so on, and then the active things, and then the lamin, and then you can look at validate that line elements of X chromosomes are co-localized. Nucleolus, as well as the nucleola RDA containing chromosomes are all co-located, so you can validate it this way. But from these set of images, what you can do is you can say, for each of the loci, each of these dots we detect in DNA FISH, right? What is the nearest immunofluorescence pixel, and what is its intensity? So you can ask whether the heterochromatic marks, are they close to any particular set of DNA loci? And so you can generate this map, right, for all 2,000 loci, that we image by DNA seqFISH Plus, and look at essentially the values of the histone marks that are associated with that loci. And this is, of course, the histone modification immunofluorescence is done at diffraction limit resolution. So it's basically, what fraction of these loci are near these marks within about 300 nanometers. So let's zoom into this particular activation mark, H3K9 acetylation for one of the chromosomes, and you can see that across this chromosome there's a profile. And what's surprising is that if you compare the ChIP-seq data that's now been at the 1 megabase resolution, so it matches with the imaging data, you can see there's actually very high concordance between these two measurements. And what's even more surprising is that if you look at individual cells, now there's about 400 cells we imaged in this particular experiment, and for along each of the cells, actually you have a fairly deterministic pattern that this loci here is touching H3K9 acetylation essentially in almost all the cells, and we call those fixed loci because they're essentially next to these marks in all the cells. This is true not only for this mark; it's true for actually almost all of the marks we looked at. So there's another example looking at the speckle localization patterns. Which loci are close to DNA loci are close to nuclear speckles based on our data with this FS splicing factor SF3 versus the Sprite data. We're looking at speckles. They're also very highly concordant and also consistent between individual cells. Now, because we got this data right from the imaging data, we can ask, where do these fixed loci, where are they actually in the image, in the nuclei? And I should mention the fixed loci are determined based on this ensemble average, essentially bulk averaged over many cells profiles. It's not determined from single cells, but let's look at them in single cells. So that's the fixed loci for the speckles are shown in purple. That's the purple dots. Those are the DNA loci. The white ones are the rest of the DNA locus. Loci are measured in that particular cell. And then the translucent pink things are the speckles. So I hope you can see that that the fixed points, which is defined across all the cells, average over all the cells, individual cells are actually coating the surface of the nuclear speckles. Same for the actual heterochromatins. So the heterochromatin is labeled in green here, translucent green, but then the fixed loci for the heterochromatins are the green dots. They're also living on the three-dimensional, on this surface of the heterochromatic globules. Similar things are seen also for nucleolus. You also see them living on the surface. And, am I just pulling out cell 38 of the 400 just because this is a special case? No, it's not. You can look at other cells. You can see, this is for a different cell, the same pattern, is there. It's just amazing that the fixed points, again, which is defined based on the ensemble measurements, right? Even in single cells are very deterministically, fixed to the coated to the surface of these nuclear bodies. Sometimes they do move away. It's not like 100% of the time. So it's a very high fraction, 90% times for many of these things. And this is just showing you some other histone marks, the acetylation mark and the K27 trimethylation marks. You have the fixed points, DNA fixed points associated with them are also very deterministically coated on this two-dimensional surface rather than 3D. So, there's sort of here's just a few other cells, but the picture is really that many of the DNA loci are not living like it's not like filling a three-dimensional space. It's like they're more like inside of a baguette, right? They're sort of this kind of two-dimensional structure that have a lot of holes in them, and the holes are actually filled with proteins and also repeat regions, that in the genome. So we have these fixed points, and if you look at individual chromosomes, you can see like some of the chromosomes will have fixed points, one part of the chromosome, and then fixed points for another nuclear body for another part of the chromosome. So in this case, chromosome 4 has this structure. The heterochromatin fixed points are at the beginning, and the speckles at the end. And if you look at where chromosome 4 is in this particular cell here, you can see that the beginning part touches the heterochromatin, the end part touches the speckle, and then the middle is sort of just jumbled around. And again, this is reproducible across many different cells. You have the parts that touches the heterochromatin and the parts that touches the speckle. Here in a different cell, you have two separate alleles, and they're doing the same thing. So the arrangement of the nuclear bodies can look very random in individual cells, and the arrangement of the chromosome can look very random. But when we put the two things together, you have this very deterministic, anchoring points with respect to each other, and they're, organized in this very deterministic fashion. So this is for a different chromosome 17 in the same cells. Again, they have this pattern that the speckles can be in some other places, but then part of the 17 always has to touch the speckle as well as the heterochromatin. And this is for the nucleolus, for chromosome 19. Again, you see the same kind of patterns. And what's actually even surprising is that if you look at what the active transcribed genes are with respect to the globule of the RNA polymerases, and we measure the active transcribed genes by intron-seq-FISH, and you see that the introns are actually, these active genes are on the surface of the RNA polymerase globules, rather than the inside of the RNA polymerases. So that's also somewhat surprising, right? Like, why would the loci actually be living on the surfaces there? And there's just a few more other examples to show you that this is seen very consistently between different cells. Okay, so this is the overall picture, that you have these anchoring points, and there are things that are very consistent between different cells, very deterministic. And because different chromosomes have different arrangement of these fixed points, right, for the different chromatin marks, that in fact you have essentially a very deterministic scaffold for the chromosomes, even though individual chromosomes look very random in individual cells. So the take-home message is this, right? Like, a lot, we think of ChIP-seq data as sort of, on one dimension, you have these peaks along the string, and you have these beads along the string. But in fact, maybe at least at the megabase resolution, this coarse grain resolution, we should think of them as sort of as DNA more coated on the surface of these two-dimensional surfaces they're living in. And then the other thing is that, having fixed points and invariances are probably the best thing we can do in biology, because, in physics, there's this theorem called Noether's theorem, which actually is probably what I would argue is one of the most important theorems in physics, because everything else derives from it. It's this relationship between invariances and conservation laws, and as well as equations, and it prescribe exactly how you get from time invariance to a Hamiltonian because of conservation of energy. And in biology, right, we're not going to get equations because the space is so big that they have 20,000 dimensions easily, right, just because you have 20,000 genes. Unlike physics, where there's essentially 4 dimensions, and so you impose some kind of invariance, the thing collapses into an equation. In biology, you're not going to get that, but this is having things that don't change either across cells or across evolution, or even could be dynamic behaviors that are conserved, are the best we can do in terms of describing the behavior. And this is because, in some sense, you can think about this as a way of reducing dimensions. Just like you can have equivalence relationships in math, right? Turns a line into a circle if you have applied the sine function, right? You can essentially, some of the points on this larger space becomes identified, then you're living on a smaller, surface. And yeah, so it's not that all possibilities are allowed, right? Only certain possibilities are allowed, and the fixed points really constrains that possibility. And I will argue that that's the kind of things we should go out and look for in various biological systems. So let me wrap up and thank the people that did all the work. And this is really very much a group effort. We had inputs from a lot of folks in the lab. And I want to really point out 2 people, Uday, who did all the chromosomal work that's shown here, and Linus, who did the RNA seq-fish plus experiment and made a lot of these things possible. And so they're the really 2 key people in the lab that really drove this method to the genomic level. And finally, I want to thank Barbara Wold, who was actually one of the pioneers of TRIPSeq and RNA-seq. And it was really this very random conversation with Barbara like a decade ago that got me thinking about how to use imaging-based method to do genomics. So thank you.
That was amazing, Wang. Thank you so much. Many questions. So I'll just start going to the audience for some of these. Michael Peppelenbosch asks, if you look at the 2 copies of a chromosome, are they spatially usually lying together, or is it random?
Yeah, it seems to be mostly random. Sometimes they're 2 separate alleles, sometimes they're fused together. I'm not sure we can really say too much about them.
Okay. Sydney Schaefer asks, is DNA relatively excluded from these nuclear bodies, or is the DNA that's in there somehow missed by your probes, like either can't bind or missed in your design or something else?
Sydney's asking a tough question. Yes, so we did some checks. We definitely fished against the repeat regions, which are usually silenced. A lot of them are silenced, and we can see that they're detected with fairly high efficiency in the heterochromatic bodies. And so we don't think it's because exclusion, because experimental artifacts is, I think the non-repetitive, I should say the non-repetitive regions seems to be living on the surfaces of nuclear bodies. And it's not, it's not an artifact in some sense, right? Because why would they also only appear on the surface of these bodies? They could live somewhere else too. So.
Well, Shaolin Wang asks, first, that's great talk. Then is asking, I'm wondering, do you observe specific pattern relative to centromeres or telomeres, and are they conserved across all chromosomes?
Yeah, the telomeres and centromeres, you can definitely see the loci are genomically close to telomeres and centromeres are always close to those things. Do we see that they have any specific pattern? I mean, the telomeres centromeres are more likely to be near these heterochromatic regions because there are also a lot of impedes near them. So I think that's all I can really say about that.
Okay. Actually, we should probably wrap up, unfortunately, and get to the next talk. I mean, fortunately, we get to the next talk, but definitely please go to the forums. I know they're.
I think we have a couple minutes, right, Megan?
Oh, we do?
Yeah.
Yeah. Oops, yeah. Sorry, we have like 2 more minutes. My bad. Which is great. So Mika Muller is asking, what happens to the fixed loci if you perturb nuclear bodies? And if these perturbations are reversible, would the spatial arrangement go back to what they were before?
Yeah, that's a great question for a follow-up experiment. I'm glad you're now a reviewer to ask us to do that question, do the experiment. But yeah, that would be a great experiment to do, like perturb the structures and see what happens.
Um, let's see. So Shen Kao asks, are the fixed points only for the genes under repression, activation, or hotspots for both?
I think so. I mean, there are fixed points for almost all the marks we looked at. So I don't know if that's, some of them are repressive, some of them are active, some of them are ambiguous. I don't know if that really answers your question.
Mike McConnell is asking, what are the challenges to applying post-mortem?
Postmortem. You mean in like postmortem human brains?
I think that is the question.
Probably what he's thinking about.
Yeah.
Go ahead, Mike.
Mike, yeah, well, send me an email.
Okay.
I got lots of complaints. But anyway.
Let's see. So Marcus Woodworth is asking, for the non-fixed loci that still have a high fraction for a histone mark, like over 50%, what do you think drives the heterogeneity of the mark? Is it like technical or is it biological?
Yeah, I mean, well, you mean like in the chromatin profile, right? Like some loci are like 90% associated with the mark, and some are 50%. That's probably not technical. I mean, because we're only looking at the loci we detected and then looking at what fraction of them are associated with marks. So I don't know. There's probably something else. Like they're not that strongly associated, and it's just stochastic then at that point.
Yeah. I mean, is it possible that it's technical with the IF part of it?
Like yeah, well, so I should say the IF is not single molecule, right? So we don't see every single histones, and so we see these globules very well. It could very well be there is some region that's looped out and it's still contacting a histone that's hanging out there that has a particular mark. So we can't say that for definite, right? But this is a global picture that's coarse-grained.
Cool. Again, Long, thank you for an awesome talk.